Hi, this is Ling from 3D Science Modeling. Many sunflowers have disc-shaped flowering heads. Have you seen sunflowers with fuzzy flowering heads? Do you know which type of flowering head is dominant? Can you figure it out? After learning about the law of segregation, students often have plenty of opportunities to use Punnett squares to analyze offspring's phenotypes and genotypes. In these practices, the dominant and recessive traits are often provided. In contrast, not many questions ask them to identify the dominant and recessive traits based on empirical evidence. Some people might assume students should have known how to do it after learning about Mendel's experiments. But the widespread misconception that the dominant trait is the most common trait in the population suggests some students might oversimplify the theoretical ratios and do not realize systematic examinations are needed to identify dominance and recessiveness. In this video, let's conduct a computer-enhanced activity to identify the dominant and recessive traits in sunflowers. The activity includes the following steps: making hypotheses, gathering data, analyze data, and constructing evidence-based arguments. A sunflower head is an inflorescence, which is made of hundreds of miniature flowers called florets. Those bright, big, quote-unquote sunflower petals are ray florets, and the small units at the disc center are disc florets. We refer to the disc-shaped sunflower inflorescences as normal type, and call the fuzzy inflorescences chrysanthemum type, because it makes the sunflower look like a chrysanthemum. For short, let's call them N-type and C-type. Scientists have found the sunflower inflorescence is controlled by one gene. In this activity, students will investigate which type of inflorescence is the dominant trait. It's not hard for students to realize they may cross sunflowers to figure out the dominant and the recessive inflorescences. The difficulty lies in how they can do it systematically and how we may support all students during the activity. Here, we use an activity sheet to guide students and hold in-class discussions at several critical learning points to bring all students towards the desired learning outcomes. We first present the photos of N-type and C-type sunflower inflorescences to students and ask them to think which type is the dominant trait and how they can test their hypotheses. This is how they do it. They first create a phenotype-genotype chart based on the hypothesis that the C-type is the dominant trait. Then, write down the possible genotypes of the parent sunflowers in different tests. Next, they use Punnett squares to predict the inheritance patterns. Essentially, students should come up with the following predictions of the phenotypic patterns for three types of tests when the C-type inflorescence is dominant. Here is the detailed process in which students use Punnett squares to make predictions. To avoid confirmation bias, students will predict the inheritance patterns if the N-type inflorescence is dominant in a similar way. In this part of the activity, students may work in small groups first, and then share their hypotheses and predictions in the whole class discussion. After all students understand the hypotheses, they're ready to gather data. Students may access the online sunflower computer model via a Chromebook or other digital device. You can find the background model information in our previous video. Students should choose case 2 for the investigation. Set up the model. Then, they may choose to cross 2 sunflowers in the model and gather data. Here, we choose 2 C-type sunflowers and cross-pollinate. We can sort the offspring to see the rough ratio and also gather accurate numerical data. We clarified to the students that they should conduct multiple trials for each type of test to gather sufficient data. Let's do it together. We'll first cross the C-type with C-type for multiple trials, then the C-type with N-type for multiple trials, and then the N-type with N-type. There's no fixed number of trials you should do. We always ask students to have more than 3 trials so that they can see all the possible patterns. We also ask them to conduct the same number of trials for each type of cross. In the present data collection, students always reset the model to gather data. If you want your students to gather data over multiple generations, such as starting with the parent generation, and then the F1 generation, F2 generation, and so on, feel free to do it. Just ensure your students understand the data collection process. Now we can analyze the data by calculating the phenotypic ratios. You can either let students use their small group dataset or compile the whole class data and then let students do the calculation. For each type of cross, we need to separate the trials where 2 phenotypes are present from the trials where only 1 phenotype is present. Very quickly, we can see when the C-type crosses with the C-type, we either get the 3:1 ratio or only get C-type sunflowers. When crossing the C-type with N-type, we either get the 1:1 ratio or only get the C-type sunflowers. When crossing N-type with N-type, we only get the N-type sunflowers. Comparing the results with the predicted pattern, now we can conclude that the C-type inflorescence is the dominant trait. From there, students may conduct evidence-based arguments. They use the results from the computer model as evidence. In the arguments, they use the law of segregation, random fertilization, the dominance concepts to reason about the evidence. Putting together an evidence-based argument is not easy. We give students in-class time to write in small groups and then share a fragment of their writing in class, so they learn from each other and receive on-time feedback. In the end, students may summarize the rules for identifying dominant and recessive traits. They may support these rules using Punnett squares. These rules can be generalized to any other characters that comply with the law of segregation and complete dominance. Some extensions of the activity include discussing why the dominant trait is not always the most common trait in the population. Students can also use the computer model to identify a homozygous dominant individual and a heterozygous dominant individual, and use the computer model data to support their claims. Find the model and activity sheets on our website. Leave your comments below. Give us a thumb up if you like it. Share the video so that more science teachers can see it. Thanks for watching.